Hello everyone, welcome to the class of T.Y.B.Sc in Microbiology for Semester 1. We have already started with the subject Enzymology, Paper 3 for Semester 1. In this video, we'll be talking about principles and methods of enzyme purification by using the property of molecular size. So before we move on to today's main topic, that is purification on the basis of molecular size, let us revise in brief about the topics covered in the previous video. So there are 3 steps in enzyme purification. The first one being solubilization of the protein or enzyme which is present in the cell. This involves steps such as homogenization, centrifugation, and filtration. Step 2 is stabilization of the enzyme of interest, and the third step is isolation of proteins or enzymes from the given mixture or cell lysate. Isolation of proteins. In our previous video, we have already talked about the first 2 steps in detail. From this video onwards, we'll be mainly talking about the third step, that is isolation of the enzymes or proteins. So methods of isolation of proteins differ from one to another depending on where the particular enzyme is located in a cell. Proteins can be isolated on the basis of properties such as solubility, molecular size, ionic charge, and binding specificity. One can use techniques such as salting out, dialysis, gel filtration or gel permutation chromatography, ion exchange chromatography, isoelectric focusing, gel electrophoresis, or affinity chromatography. The first technique which we are going to see when we talk about separation of enzymes on basis of molecular size is dialysis. Dialysis comes from a Greek word dialysis, which means dissolution. Dia meaning lysis, luisis meaning losing. Dialysis is the process of separating molecules in a solution by difference in their rate of diffusion through a semi-permeable membrane. The most common application of dialysis is removal of unwanted small molecules such as salts, reducing agents, dyes from large macromolecules such as proteins, DNA, polysaccharides which are present in the cell lysate after the process of homogenization and centrifugation. Dialysis can also be commonly used for buffer exchange and drug delivery systems. Let us move on to the principle of dialysis. Diffusion is the random movement of molecules in a solution. This diffusion is generally associated with Brownian movement of solute or solvent molecules in the solution. This movement of molecules from area of higher concentration to an area of lower concentration until an equilibrium is reached is the main principle on which the technique of dialysis lies. So in the dialysis technique, a sample and a buffer solution is required. The buffer solution is also known as dialysate. The sample and the buffer are separated by the help of a semi-permeable membrane. This leads to differential diffusion patterns, thereby permitting the separation of molecules in both sample as well as the buffer. Owing to the small pore size of the membrane and the larger molecules which are present inside the dialysis bag, the larger molecules cannot pass through the membrane and the small molecules freely diffuse across the membrane to obtain equilibrium, hence bringing about separation of solute. So this is diagram of dialysis. Enzymes can be separated from small molecules such as salts by dialysis through a semi-permeable membrane. This semi-permeable membrane can be made up of materials such as cellulose and has small pores in it. As you can see in the diagram, there are two types of molecules with difference in molecular size. The red one being the smaller molecules which have a pore size smaller than the pore size of the cellulose membrane, whereas the blue colored molecules have a molecular size greater than that of the pore size of the cellular membrane. So the proteins placed inside the dialysis bag is then submerged in a buffer solution. Molecules having dimensions greater than the pore size of the membrane are retained inside the dialysis bag. So as you can see in the B part of the diagram, even at equilibrium, the blue colored molecules which are having a greater molecular size are retained inside the dialysis bag. On the contrary, the smaller molecules and ions which are capable of passing through the pores of the cellular membrane diffuse down their concentration gradient and emerge in the solution outside the bag. The second technique which we use for separation of enzymes or proteins on basis of molecular size is size exclusion chromatography. Size exclusion chromatography is also known as molecular exclusion chromatography or gel filtration chromatography. It is also known as molecular sieve chromatography. So, as I said earlier, it separates enzymes mainly on the basis of their size. It has two phases, a stationary phase and a mobile phase. The stationary phase consists of porous beads. For example, cephadics can be there, cepharos can also be present inside the chromatography column. Whereas the mobile phase usually contains a buffer. Common examples of buffers used in gel filtration or size exclusion chromatography are tetrahydrofuran, chloroform, dimethyl formide, etc. Let us now move on to the principle of gel filtration chromatography. So as you can see in the diagram, the beads are made up of an insoluble but a highly hydrated polymer. This polymer can be made of materials such as dextrane, agarose or polysaccharides. The sample is applied at the top of the column consisting of porous beads in the buffer. So as you can see in the diagram, the small molecules of the sample can enter the beads as indicated by red color in this diagram, but the large ones cannot. So as you can see, because the yellow colored molecule is Larger in size, it cannot pass through the resin and it will be eluted in the buffer and cannot enter the resin bead. So small molecules are distributed inside the beads and between them, whereas the large molecules are only in the solution between the beads. Due to this, the large molecules are free and they flow more rapidly through this column, whereas the small molecules take a longer path and will leave last. So this is again a diagram showing gel filtration chromatography. You can see the different stages of how proteins are separated. The bead or resin shown in grey color is made up of polymer beads. You have three different kind of molecules indicating small, intermediate and large size. So large molecules, as they cannot enter inside the bead, will be eluted the first inside the test tube. Larger proteins emerge from the column somewhat sooner than the small ones. This is a counterintuitive result. The solid phase, that is the beads, are known to be consisting of cross-linked polymers. These beads are engineered pores or cavities which are of a specific size. Large proteins cannot enter the cavities and so take a short and rapid path through the column around the beads. Small particles enter the cavities and are slowed by their more labyrinth path through the column. The next technique is ultrafiltration. Ultrafiltration technique is used for protein concentration in which water or low molecular weight material are removed from the protein by passing it through a membrane in an inert atmosphere and under pressure. One can also subject the cells to centrifugation. The protein is retained and does not pass through the membrane. This leads to its increased concentration, but there is little loss of enzyme activity. The membranes with molecular weight cut off from 1000 to 100,000 kilodaltons are available, which can be used under pressure of up to 2 micropascals. So as you can see in the diagram, the protein solution is contained in a cylinder stirred cell where the membrane is placed at the base of the cell. This cell is equipped with a magnetic stirrer. An inert gas, very frequently used nitrogen, is passed continuously through the stirrer, which can force the water and low molecular weight materials through the membrane. Ultrafiltration procedures for concentration of laboratory columns, elutes, are commonly used. However, their large-scale use is not much explored, but they are very popular for concentration of solution. With this, we finish the three techniques used in purification of enzymes using the property of molecular size. In our next video, we'll be mainly discussing purification of enzymes on the basis of their solubility. These are the references. Thank you so much.